Hey guys, welcome back to the second part of Chapter 6. We're going to continue our conversation about the sensory system. So last that we spoke, we were talking about touch, we were talking about proprioceptors, we were talking about different mechanoreceptors. So let's go ahead and continue that trend, and now let's focus on equilibrium and hearing. All right, now both equilibrium and hearing are examples of mechanoreceptors. And what we see happening is that when you talk about those terms, equilibrium and hearing, you're discussing two different sensations. Equilibrium, some people like to describe as balance. It's basically how our body is able to react to the force of gravity being expelled on us. So are you able to walk up straight? Are you leaning? Are you falling over? Hearing obviously means that you're able to not only detect, but you're also able to interpret sound waves that are heading your way. So the fact that as you're walking on this road, if you're walking on the street, you hear an ambulance, that you're able to quickly detect where it's coming from, and you kind of step aside out of the way. In vertebrates, what we see is that the ear will be responsible for taking care of both our equilibrium as well as our hearing demands. Whereas in vertebra, they will have different organs for each. And in fact, speaking about the invertebrates, because they'll have a different organ for their equilibrium and one for their hearing, what we see happening is that they can go ahead and be very specialized in their entities. So here you have an illustration of a lobster and an octopus, and it's showing you that both of them have this organ called a statocyst. And a statocyst is going to be a specialized organ that will completely focus on equilibrium within these invertebrate models. It is based on the fact, and you can see it at the bottom of the illustration, that it will have a hollow cavity, and the cavity will be lined around its entire perimeter with ciliated sensory neuron cells. And these are all examples of mechanoreceptors. And inside of the cavity, you're going to have these little particles of calcium carbonates that we're going to call statoliths. And what we see happening, for instance, with the lobster is as the lobster is walking around, the statoliths basically move in the cavity, and the cavity, as it's moving within the cavity, it's interacting with the sensory neurons, thereby able to detect if the lobster is moving in a forward, backward, side to side, straight up type of pattern. It's actually a very simplistic method, but it is very effective. The octopus takes it a step further. In his statocyst, what we see happening is he will also utilize the statoliths, but he will go ahead and have different options as to how these calcium carbonates can interact with the neuron cells. So on the picture of the octopus at the bottom, you're going to notice that you have the different cristas and you have the macula. These are all different variations of branches of the statoliths. Inside of each, you'll have your sensory neurons, and what we see happening, because the octopus has a variety of cristas and a macula, it is able to give a more detailed analysis of its equilibrium and its the position of the body as it comes to gravity. So for instance, the macula will go ahead and focus on if the animal is moving forwards or backwards, right? But the cristas, they are going to be able to detect any type of angular acceleration, so any type of turning that we can do. And believe it or not, octopus, they're actually capable of doing very complex behaviors, and there's been examples where they have created mazes for them, and they've been gone ahead and detected from beginning to end, even doing complex flips along the way, and that's because they're able to have such control over their equilibrium. Sticking with the insects, their hearing is going to be more done or controlled by our ever famous sensilla. We're going to use lots of trichoid sensilla, and as we were coming towards the end of part 1 of our chapter 6 lecture, I did mention the fact that they can also utilize the tympanal organs. They are able to have an increased sensitivity, part of that because of its interaction with the cuticle, and it has a very thin layer that overlays the chorotonal organ. I see I kind of skipped something along the way, my apologies. So in most insect models, what we see happening is that sound is controlled by the chorotonal organs. And the chorotonal organs is a variation of the sensilla, because it will go ahead and just cluster together, and usually it's located around the leg, and because it's a mechanoreceptor, we see that it will have changes in the opening and closing of the ion channels. The problem with the chorotonal organ is that it's not very sensitive. So it can detect the sound, but it doesn't really tell you if it's a loud sound, a small sound, a light sound, where exactly it's coming from. If the insect wants to have more sensitivity, then it's going to have to rely on the tympanoid organ. And what we see happening over here is because it has that thin layer of cuticle that crosses over the chorotonal organ, that cuticle is almost going to act like a little drum. So as the vibrations and the sound waves come in, it basically starts interacting, causing vibrations on the cuticle, and this cuticle is then able to amplify the sound and able to go ahead and quickly communicate that with the nervous system. So that's why the tympanoid organ is more sensitive than the good old-fashioned chorotonal organ when it comes to insect hearing. In vertebrates, what we see happening is that we are going to utilize hair cells for the ability to do both of our hearing and our balances. I also want to point out that the hair cells, in addition to doing both of these functions, they are also modified epithelium cells. So we're not utilizing neurons, we're doing modified epithelium cells, just like we did when we were talking about taste. Now go ahead and take a look at your picture of your typical hair cell, and what you're going to notice is that you have stereocilia on the side connected through a little tip link, and then you're going to have an elongated cilium. This is going to be your kinocilium. And what we see happening is that your stereocilia will be the ones that will be detecting the wavelengths as they come along, and they're able to move either towards the kinocilium or away from the kinocilium. And we can then use that mechanism to detect if the sound is coming towards us or away from us. So for instance, usually if there's a vibration or a sound wave that's getting closer and closer, what we see happening is that the stereocilia will be able to detect it, and they'll pivot towards the kinocilium, thereby opening lots of ion channels and creating that membrane potential, alerting the nervous system that the sound is coming near us. As the sound is getting further and further away and becomes harder to interpret, we see that the stereocilia will start moving away from the kinocilium, and by that it will go ahead and induce repolarization, allowing the membrane to reset back to its normal non-stimulated setting. We also see that if we were to study hair cells in fish and amphibians, that oftentimes they're going to go ahead and they're going to modify it a little bit by creating these areas called neuromasts. Now, the neuromasts, I'm going to show you a picture of it in just 1 second. It's basically going to be a combination of hair cells sitting in a little gelatin cup that's called cupula. It's going to be filled with a jello-type liquid, and it's going to be utilized to detect the motion of the water as the fish is moving through its environment. Most of us, if we're not familiar or if we've never heard from the neuromast, we have heard of a lateral line. We've definitely seen it on some of the fish that we've been exposed to. And in fact, go ahead and take a look at your illustration, and you're going to notice that you have this line that runs the entire length of the body. That's the lateral line. You can see it on our little dogfish over here as well. And what the lateral line is nothing but just a singular arrangement of neuromasts. In fact, let me go ahead and take care of this picture so we can see our neuromast. Here we go. So if we were to, let me just get rid of this red line. Give me 1 second, please. This is what your neuromast looks like. And as I said to you, it's going to be a collection of your hair cells. So here are your hair cells in close communication with your neurons so they can detect and reflect when they're stimulated. And now what we see happening is that they're encapsulated in a little cup. We call it a cupula. and the copula is full of like a jello type liquid. And what we see happening is as the fish is moving through the water, it is able to cause vibrations and allow the fish to detect any type of changes in the water pressure or water movement. So it can detect, for instance, if there is a predator that's coming from behind it. Now back to our vertebrates. So here's an illustration of a typical vertebrate ear, and what we see happening is that it's divided into three sections. And a disclaimer, not all vertebrates need to have all three sections to be successful in both hearing and equilibrium. So the three sections are going to be an outer ear, a middle ear, and the inner ear. And out of the three parts, the only part that everybody needs to have needs to be the inner ear. And the reason for that is because that is where you're going to be housing your hair cells. So those are the cells that are able to take the vibrations and communicate that with the nervous system. The outer ear is basically going to be your earlobe, which is more scientifically called your pinna. And the larger the pinna is, the actually better it is because it tends to amplify the sound. I don't know if you guys have ever done this. If you're playing a song on your phone, maybe you forgot your Bluetooth speaker, what you can do is you can take your phone and you can put it in a cup or in a glass, and you're going to notice that it amplifies the sound. And the reason for that is very similar to how the pinna or the earlobe works. It's able to catch the sound and basically just surround it, thereby allowing it to be amplified. And as it's being amplified, it will start traveling into the auditory canal, and that is also part of the outer ear, and eventually it will make its way over to the middle ear where it will encounter the tympanic membrane. And the tympanic membrane, very similar to a drum, will start vibrating as the sounds get closer and cause the malleus, the incus, and the stapes, these three small little bones in the middle ears, to vibrate as well until it can take the sensation and move it over to the inner ear where we're going to have our cochlea. Kind of looks like a coiled up snail. The cochlea is where you have your hair cells that are housed, so where the stimulus can actually be interpreted and you can get your membrane potential change that you're looking for. Here's a look at how fish are able to do it. Fish will actually also utilize their audioliths, those little calcium carbonates that we see in the Statocysts that we were chit-chatting about before in like lobsters and an octopus. And either way, what we see happening is when it's able to detect sound, we see that the swim bladder of the fish will also play a role in amplifying its interpretation of it, because sound will tend to vibrate off the bladder and it will pass over and will start interacting with the webinar ossicles, which are like a little collection series of bones. They are able to connect the bladder and the ear, thereby able to amplify the sound that's being interpreted. Now back to our little inner ear, what we see happening is that if we uncoil the cochlea, that is where all the magic is going to happen. And I have to apologize because the inner ear is going to come up in our next slide where we can take a look at where the hair cells are exactly. This picture on this particular slide is meant to highlight the middle ear and the fact that you have three little bones in there, the malleus, the incus, and stapes. Please know these bones in order and know that the malleus is the ones that will connect with the tympanic membrane, and the stapes is the one that will connect with the cochlea, okay, the coiled up section right over here. And this is all meant in a way to amplify the sound as it's traveling through the auditory tunnel on its way to the tympanic membrane where it beats up the drum, and then it goes ahead and it gets amplified through the vibration of the malleus, the incus, and the stapes. And like I said to you before, not all vertebrates need to have an outer and a middle ear, but all of them will need to have the cochlea, which is located in the inner ear. And this, of course, is going to be because of the formation or the location of the hair cells. All right, here's the picture that I was looking for previously. So here's my cochlea, and as you can see, It has started to uncoil itself so that we can better see what's happening. The sound has entered through the auditory canal. It has vibrated on the tympanic membrane. It has traveled through the malleus, the incus, and the stapes, and now it will encounter the oval window. And as it goes into the cochlea, what we see happening is that it's going to be exposed to two different sections. It's going to be exposed to the vestibular duct and the tympanic duct. And I'll just go ahead and draw my arrow so you can see the different ducts. Now, the vestibular ducts and the tympanic ducts will be filled with fluid. And this fluid is called perilymph, and it is a combination of high sodium, low potassium. Very similar to the combination of ions that you would come across in the extracellular fluids. And as the sound passes to the vestibular duct, it will start to interact with the basilar membrane, the tectorial membrane, and the hair cells that are located in the cochlear duct. And the cochlear duct also has a fluid. It has endolymph fluid. That one is going to be the opposite of the perilymph. It's going to be high in potassium and low in sodium. And the alternation of these ions allow the hair cells to become excitable from the sound waves that are traveling in it. And we see that the hair cells will form an inner and an outer row. The inner row tends to be more for sound detection, whereas the outer row is all about amplifying and sort of like clearing up the sound to make it easier to analyze. Beyond that, once the hair cells are activated, we can go ahead and see that they'll have a clear line of communication with the nervous system. So you'll have the opening and closing of the ion channels, the change in the membrane potential, and the passing on of the signal to the integrating center. Our next category of receptors will be photoreceptors. Photoreceptors are the ability to detect photons or visible light. We are not able to detect all the colors. We can only see a small portion of the light rays that are available up there. I think humans are between 350 and 750 nanometers. So that's our little range. That's the one that we are detectable with. Oh, it's right over here on the PowerPoint. Here you go. And it turns out that visible light travels well in water. Other wavelengths do not. And they kind of utilize that to kind of see from the fact that most likely animals evolved sight initially in water before they migrated onto land and further evolved that particular sense. All right, now when it comes over to photoreceptors, what we see happening is that it can be a range of just having like a very simplistic cell to having more complex organs such as our eyes that are able to detect lots of details, lots of contrast, lots of different colors. But basically what we see is that we're going to have two major types of photoreceptor cells. For the vertebrates, it's going to be the ciliary photoreceptors, which is going to have highly folded cilium and can form a disc that contain the photopigments. Don't worry, you'll have lots of pictures that'll come your way that'll describe this. For the invertebrates, they're going to go ahead and rely more on the rhabdomoric photoreceptors. This one tends to be more of an open apical surface that will have little microphyllae projections that will be outfolded. And within these projections, that is then where you're going to have your photopigments. Your photopigments are the ones that are able to detect the photons and cause the sensory cell to respond to them accordingly. Here is a look at the vertebrate's photoreceptors, and please keep in mind that you tend to have two variations of them. You tend to have your rods, which are specifically aimed at dim light, and you have your cones, which are more specialized for bright light reception. So for instance, if we were to look at vertebrates that tend to be nocturnal, they will have a high amount of rod cells available in the eye. Whereas when we look at humans that are able to see both, but we're not really nocturnal, we usually have a combination of rods and cones. From the anatomy standpoint, you can see that the photoreceptor has an inner segment and an outer segment, and the outer segment is where you would go ahead and be able to contain your photopigments, and the inner segment is then where you're going to go ahead and be able to make your connection with the other neurons that will communicate it to the sensory receptors. All right, so let's take a look at all the different ways that an eye can form in different organisms. And the importance of an eye is that it allows you to have a structure that can actually detect the direction that light is coming from. So you can have a very simplistic structure, just a group of cells. We could call it an eye spot. And this is what you see, for instance, in euglena. So you can have it here on your picture. You see this little red dot right here? That is an example of an eye spot. It basically means that you have a group of cells that are able to respond to photosensitive pigment. And then what the organism sees is it actually can just see a light coming towards its way. Nothing super fancy, but it can see if a light is coming towards it. An eye, on the other hand, is going to be classified more as an organ because in addition to being able to detect light, it will also allow us to kind of contrast where the stimulus is coming from. And we can also utilize the expression of colors and resolution, so sharpness, so we can utilize it to form images. There are different types of eyes, and it will all depend on how the photoreceptors are aligned together and are interacting with each other. So for instance, our first example is going to be what we like to call a flat sheet eye. This is usually what you tend to see in larval forms. There's also some adult forms, but most of these tends to be in the larval forms of organisms. Now, in a flat eye, what that means is that the photoreceptor layers are just literally lined up in one line. They'll have a pigment layer that they'll interact towards the bottom that can communicate with the afferent neurons. Flat eyes are good at giving a sense of light direction. So is it coming towards it or away from it? And it can also kind of see if it's a bright light or a dim light. The cup-shaped eyes are the next one. This is when you have a retinal sheet that will start to fold and it will surround, it will form a narrow aperture. This one is good because of the formation of the retina. We see that you have more of a light-dark contrast, and that will allow you to focus your photons so that they can quickly interact with the photoreceptor cells and the afferent neurons. The cup-shaped eyes will allow for an image to start forming, but it will be very poorly resoluted, meaning it won't be very sharp. It will almost be kind of like a vibrating dot. That is coming your way. So you can discriminate between light and dark, and you can see the darkened region moving and vibrating, and that is the image that's starting to form. If you want to go ahead and see with great detail and have lots of good resolution, then what you need to aim for is a fascicular eye, which is what we have. The fascicular eye means that in addition to the retina, we're also going to incorporate a lens, and the lens is going to allow us to focus the photons that we're receiving so that we can have improved clarity and intensity. It's also going to allow us to reflect the light so that we can go ahead and quickly communicate the signal with the photoreceptors. And because of all this additional input and customization, we see that we can get a beautiful picture, fully in color, and able to have lots of details that we can interpret from it. If we want to take the concept and kind of do an outward radiation of the photoreceptors, then we call that a convex eye. And that's what you would see, for instance, in annelids, arthropod, and mollusk. And you would also have a convex retina because, once again, you are dialing outwards, not inwards. We also see that within the convex eyes of the arthropod, we see that they tend to be composed of ommatidia. Ommatidia are small little sections that will each have their own little lens. And what you can do is you can go ahead and take a look at the illustration that's right over here, which is a zoomed in analysis of our little fly. And we can see the different little pieces that make up the convex eye, and this is then the ommatinia. So all these little segments that you're seeing. So you can have hundreds, if not thousands of them present on the arthropod, and each of them has its own lens, which means, and you can see it on the illustration right here, all these little dots, each of them is able to take in a photon and analyze the image. The question then becomes, what do we do with all this individual data? How do we put it together to form our image? Well, you can have two options. You can do an appositional compound eye, which means that each ommatiya operates independently, meaning that it will cover a different part of the image. And then you'll have your neurons that will merge the image together. Or you can do the opposite, which is the superposition ommatiya, which basically means that collectively they'll always work together to merge one full image. So in one, it's almost like you're cutting and pasting to form the image, whereas in the second scenario, all of them collectively work together. We see that by having multiple ommatiya, what does happen is that you have an increased amount of resolution that can happen. Excuse me. And it also allows the arthropod to have a wider array of sight. So it can see more of its environment within the same setting. Let's take a look at a few structures that you would come across if you were to ever dissect a cow's eye or an invertebrate's eye. Or in the case for those of us who won't be dissecting this semester, we can go ahead and just take out a mirror and take a look at our own eyes, and we should be able to identify most of the key terms that are listed on the PowerPoint. The first thing is that when you look at your eye in the mirror, you're going to notice that you have a lot of white towards the front. All that white is part of the sclera layer. This is a connective tissue layer that makes up the superficial layer of the eyeball. Towards the front of the eye, where you have like the actual coloration of the eye and you can see your little pupil, you have a little transparent layer called the cornea that sits over it as an extra layer of protection. Within the eye itself, you're going to come across two additional layers. You will have your retina layer, which is your innermost layer. This layer is the one that will have all your different cones and rods. So this is where all your photoreceptor cells are located. And then the middle layer is going to be called the choroid or choroid coat. This is a pigmented layer. As you can see on your PowerPoint, it says it lies behind the retina. Some of the animals who have more of a nocturnal type of lifestyle, they will also have on top of the choroid, they will have a tapetum layer. And this layer tends to be a beautiful fluorescent color. It basically allows the animal's eyes to kind of reflect light during nighttime. This is also one of the reasons why if you've taken a picture, for instance, of your cat or your dog with the flash on, when the picture kind of comes up, it has that nasty red color that kind of reflects from their eye. That is the choroid and the tapetum coat reflecting back. Our choroid coat does not have a beautiful fluorescent color to it. And the color, I'm going to show it to you in 1 second when that picture pops up, I promise. But either way, I definitely wanted to highlight the fact that the eye is going to be composed of 3 layers. The sclera on top. In the middle, you're going to have your choroid or choroid coat. And then the innermost layer is going to be the retina. Now back towards the front of the eye, you're going to notice that your eye color is due to what we call the iris. The iris is actually a pigmented smooth muscle. It's important to keep that in mind. Because it is a smooth muscle, that will mean that it can dilate and it can constrict. And as it's dilating and constricting, you're going to notice that the little black dot in the middle of the eye gets wider and smaller. Now, the actual opening that's exposing that little dot, that is called the pupil. So on your PowerPoint, it says the pupil is the opening in the iris that allows light into the eye. And the little black dot that you're seeing, that's actually the lens. The lens almost looks like a little marble. It is held in place by these thin little ciliary body strings, and it will allow the lens to kind of change shape as the pupil opens and closes to allow light in, so that the lens can go ahead and take the images received and reflect it back on the retina coat behind it, so it can obviously go ahead and communicate with the proper photoreceptors. Now, in case you're wondering, there's also lots of liquid in the eye itself. The eyeball is full of viscerous humerus, which is more of like a jelly type of liquid. Unfortunately, as we start to age, the jelly starts to liquefy and becomes more water-like. That is one of the main reasons why naturally, as you get older, you do tend to lose some of the quality of your eyesight, because it makes it harder for the lens to communicate with the retina and get those images nice and clear. Towards the front, by the anterior chamber, where your iris, your pupil, and your cornea are located, you also have a little bit of liquid. This looks almost like water. It's called aqueous humerus, and this is just a few drops just in front to kind of lubricate everything that's towards the anterior of the eyeball. Let me go ahead and have that picture come in. Here's our little picture that popped up. Now on this one, what we're looking at is basically the back half of an eyeball. The front part has been cut open, and if you bear with me on the next slide, you'll be able to see an illustrated eye with all the different parts mentioned. But I like this picture because it's going to be able to highlight for us the three main sections of the eyeball, or I should say the layer. So the white that's on the outside, the first one, that's going to be your sclera. And then this beautiful black and luminescence coat, this is going to be our choroid layer. Now, the fact that you have the fluorescent binding, that's the tapetumum that we were mentioning before. So our eyes will not have that fluorescent area because we're not classified as nocturnal. Ours will just be a simple black layer choroid coat. And then the last one is this part right here. Now, this layer has been removed just to kind of show you more of the choroid coat. And this layer right here, this is the retina layer. So this is your innermost layer. This is where all your photoreceptors are. And even though it's not that great to see it here, the layer is actually attached towards the back right here by the optic disc. And the optic disc on the inside is also called your blind spot. It will correspond perfectly with the optic nerve that's on the outside of the eyeball, so it can communicate any of the signals received from the sensory photoreceptors over to the brain, so the image can be analyzed by the occipital lobe. If we come over to this picture, we're able to see a lot more of the items that we were discussing before. So for instance, you can see your iris. The iris is the color of your eye. It's composed of muscle, so it can constrict and dilate, and as it does so, it controls the opening right here, which is called the pupil, and that will determine how much light is able to enter into the eye and interact with the lens. Here you go. And as you can see, the lens is being propped up and kept in place by the ciliary body. So as the photons come in, the lens will go ahead and start moving, and they'll be able to communicate it directly with the retina in the back, which will then go ahead and stimulate the cells to communicate it through the optic disc and the optic nerve, thereby allowing it to connect to the brain and the rest of the nervous system. Now over here it says that in order for us to get a clear form of an image and to have as much detail as possible, we do see that we're able to do refraction, which basically means that the cornea and the lens will start focusing light on the retina by altering their location, so there'll be a slight shift. And then what else do we see? We see that the lens can change shape to focus on nearer for objects. That's called accommodation. So if you are nearsighted or farsighted, usually what we see is that your lens has some issues with accommodations, so you might need to correct that with some glasses or contact lenses. All right, now continuing off with the retina, as I mentioned to you before, that is where the rods and the cones are going to be located. So as the light enters here, the lens will move itself, so it makes it easier for it to reflect it over the photons to the retina. And what we see happening is once it starts interacting with the retina, it will have an easy time connecting with the bipolar cells and the ganglion cells as it's making its way over to the optic nerve. Now, the area where the optic nerve exits the eye is called the optic disc. And the optic disc is also known as your blind spot, because that particular section does not have any cones or rods to interpret. So if you've ever done the blind spot experiment, and if you're in lab, you'll do one at home that you'll be able to see, where we take anything as simple as a sheet of paper, we'll put a little plus sign on it, and what we see is, depending on how far the paper is away from our eye, we're going to notice that for a millisecond it will disappear before it comes back. And when it disappears, that is when the image was reflected on the optic disc, the blind spot, and you were not able to interpret it. The fovea is the region where you have the sharpest image, and part of that will come from the fact that that area is full of cones. So lots of color vision. This is the center of the retina. So what we see happening is, because of its overall location, it's quickly able to interpret an image and give you a nice contrast of it. So it's the complete opposite of your optic disc, which is your blind spot. The other thing to keep in mind when you talk about vision is the fact that because we have two eyes, what we're doing is we're doing binocular vision. Binocular vision basically means that we can take the individual visual field of the eye and we cause them to overlap, so that we can have a wider array of our environment that we can see simultaneously. And it turns out by doing that, it also allows us to create more of a three-dimensional environment that we're able to observe. So this will have to do with issues like high amounts of resolution, but more importantly, depth perception. So it allows us to really analyze how far an object is from us, and if that object is flat or has more of a three-dimensional shape. I also want to point something out that's more towards the top of the text in the PowerPoint, and this basically shows you that when the stimulus comes in, the photons comes in and it interacts with the eyeball and it interacts with the retina. What we see happening is, once those cells are all activated, the stimulus will go ahead and exit through the optic nerve. Once it's in the optic nerve, it will travel through this little X cross section that is called the optic chiasma. And when it does that, look what happens to the stimulus. Anything that was on the left will now cross over to the right side of the brain. Anything that was on the right will now be crossed over to the left side of the brain for analysis. And it will travel its way down the optic tract until it's able to go ahead and communicate with the visual cortex that's more towards the occipital lobe, of the brain. So the optic chiasma is a cross, that little X, and it allows us to flip the stimulus and the reception of the brain from one side to the next. So anything you see on the left is interpreted on the right side, and anything that you see on the right is interpreted on the left side of your visual cortex. One last thing about vision is our color vision. This will all be determined about depending on how you're able to detect different wavelengths of visible light. Most mammals can only really do two colors. They tend to focus on blue and red. Humans, we are trichromatic, meaning that we can see two. We can see blue, we can see green, and we can see red. And what we can do is we can combine those colors, obviously, to make a wider combination of colors that we're able to interpret. We're not as good as some birds, reptiles, and fish, because they're able to become tetrachromatic or pentachromatic, meaning that they can have a much wider degree of discrimination between the colors that are available. So they're really able to determine if you're looking at a light, medium, dark color of blue, green, or red, and anything in between. So it's excellent for better color discrimination as well as UV detection. Now, finishing off our chapter, we just have our thermoreceptors to chit-chat about and our magnetoreceptors. Thermoreceptors are going to be all about temperature changes, and we basically have two categories to look at. We have the one that's going to be involved with our internal temperature and communicating it with the hypothalamus. Those are going to be our central thermoreceptors. And then we have the ones that will monitor the environment around us. Those will be our peripheral thermoreceptors. The peripheral thermoreceptors are the ones that are going to tell you that the water in the hot tub is hot. Or if you pick up an ice cube because you dropped it, that's cold. So it's able to monitor our external environment that we are exposing our skin and our body to. We also have presence in some mammals of what we call thermoreceptors. These are specialized peripheral receptors that are able to detect stimuli that are above 45 degrees Celsius. This could be obviously very detrimental to an organism if you expose yourself to that amount of heat. All of these thermoreceptors are going to be utilizing a transient receptor potential ion channel, a TRP, when they become activated, and they're able to communicate it with the thermoreceptor proteins within their proper receptors. I also want to point out of the little picture that we see of our snake right there, is that in some animals, primarily snakes, we see that they're able to have what we call a pit organ. A pit organ is going to be an example of a thermoreceptor that's going to be able to detect extremely minute changes in temperature. In fact, what you can do is go ahead and take a look at your column on the right, and it says that pit organs, which are located within the eye and the nostril of this viper right over here, they can detect 0.003 degree changes in Celsius. That is an extremely small change in temperature. And if you don't believe me, go ahead and look what I wrote for you in parentheses. Humans on their best day can only detect about a half a degree of change in temperature. So these pit organs are fantastic at being able to detect any type of heat that's radiating from an object. And our last one are magnetoreceptors. And I'm sorry to tell you this, but we don't classify for them. Magnetoreceptors are the ones that are able to detect a magnetic fields. Unfortunately, humans lack these. We do see it in a lot of animal species, especially those that like to migrate. So I wrote down migratory bird, the homing salmon. There's also been cases of seeing it in lobsters. And basically what's happening with the magnetoreceptors is that they tend to have a sort of natural mineral arrangement, very similar to almost like the compass of a needle, that is able to detect and respond to any magnetic field, and they can utilize it almost as a magnetic symbol, so they can figure out where they have been and how to get back to that precise location. So that's very interesting, and as I said to you before, it comes in very handy, especially when you look at migratory patterns of different animal models. That is the end of chapter 6. I am very excited for our next chapter. That one's going to take a look at the nervous system, and we'll be able to explore the different parts of the brain and see what each part contributes. And of course, a main purpose of getting the nervous system after this chapter is because all of the information that our sensory receptors were able to detect and transduce and have their ion channels respond to, causing a memory potential, all that information was sent down to the nervous system. So it would only make sense to now figure out what exactly are we going to do with all that information that has just been received. All right, so having said that, let's go ahead and finish off chapter 6. And like a broken record, I have to remind you, any questions, comments, or concern, please feel free to email me. All right, you guys enjoy the rest of your day, and we will talk soon when we go ahead and do the nervous system, also known as chapter 7.